So we just talked about transition and transversion. So this is probably you also learned it from like earlier class, but we want to sort of remind you the transition is between A and G or between T and C. Then the transversion is between purine and pyrimidine. So that's the different type. And this is related to the mutation rate, which we'll talk about when we start talking about the models of the substitution rate. And we'll talk about the rate, like differences between transition and transversion. And the other two here is the synonymous and non-synonymous. So I already briefly talked about synonymous. We also call it silent changes. So the definition for synonymous, the change at the DNA sequence level doesn't affect the protein sequences. So this is the definition for synonymous. So the change at the DNA sequence still encode the same protein. Then we call it synonymous changes. Non-synonymous is on the other way, like opposite meaning, because the sequence change at the nucleotide level also change the protein sequence. And if you think it's not easy to say synonymous and non-synonymous because it's a long word, you can use silent and replacement. Actually, a lot of people use replacement to say non-synonymous side. It's interchangeable. So it's silent mutation, replacement mutations. So the synonymous and non-synonymous. And here just an example to show you the change. So at a DNA sequence, I just put the messenger RNA here and the amino acid sequence at the bottom. So if the sequence do change, like the sequence at nucleotide level do change the amino acid sequences, then you will see the differences. So this is from here to there. So this is the first, like just assume it's an ancestral sequence, or just take an example of this codon. It's a CCG, and if the second position changes to T, that's the second column here. You see the leucine. Then if the last, from leucine to the last codon, they change at the third position and doesn't change the amino acid, so it's still leucine, right? So you can see for the first two columns, if change from proline to leucine, they'll say that's the non-synonymous change, or that's a replacement. And if from leucine to leucine, although the sequence change at the third position of this particular codon, but the amino acid is still the same. So that's the synonymous change or silent mutations. And this is just a sort of like a repeating slide, but to put it in a different names, but it's still the same. So the replacement changes, the silent mutations, and also the examples here. Then the indels, so we already saw the indels in a stretch of sequence in a couple of slides before this. And this one just talk about at the coding sequence, if you have non-triplet insertion or deletion. Now it has a non-triplet because our codons are like counted by 3, right? So 3 nucleotide will encode one particular amino acid. But if you have, say, one deletion occur here, then the rest of the sequence will shift. Then we call the friend shift. In coding sequence, we have coding friends. So if you look at it like NCBI, look at the gene bank, a sequence will tell you the coding friend. If you give a stretch of DNA sequence, there are 6 different coding possibilities, 6 friends, like starting from position 1, 2, 3, or -1, -2, or -3. So that's 6 different friends. But for a given gene, usually only one reading frame is the correct one. So when you have one base pair or non-triplet indel insertion or deletions, then you will have a frame shift mutations. So the coding frame will shift and usually will cause a stop codon. So this is just give you a long stretch of sequence. I think you don't have any problem to recognize synonymous and non-synonymous. If in our midterm, I'll give you the codon table so you don't have to memorize it. But I will ask you certain questions regarding whether this particular one is a synonymous or non-synonymous, or we call it missense. This sense is a different one. It's the non-synonymous change, a different terminology for non-synonymous change. We also say we can call it replacement. So this can be easily recognized by provide a codon table in hand. You can easily check it. But there's also a possibility, for example, we have a nonsense mutation. A nonsense mutation doesn't encode anything. That means stop codon. So in our codon table, in most eukaryotic codon table, we have 3 genetic codes encode stop codon. So that's the termination signal for translation. And this is an example of nonsense mutation. If we have a nonsense mutation, then it's like all the sequences after the stop codon will not be translated. So even it still contains good reading frame, but nothing will happen after the stop codon. So that's the nonsense mutation. So recombination. In your genetics book, most of the time we talk about recombination, we're talking about the crossing over occur at the chromosomal level. So this is what you learn from genetics. But there's the other type of recombination concerning about gene conversion. And we also sometimes say this is a non-reciprocal recombination. So when we say reciprocal recombination, the crossing over is the 2 homologous chromosomes exchange materials. And that's the figure you can observe in your textbook. But when we talk about gene conversion, it's only one particular chromosome changed and based on the other homologous sequence. So that's why we say it's a non-reciprocal recombination. So there are 2 types of recombinations at the sequence level. Deletion, insertion, I already mentioned. Usually we don't know it's insertion and deletion, but I didn't say, but the terminology now we combined insertion and deletion to a new word, indel. A lot of the papers in molecular evolution, we use indel directly because we don't know whether it's an insertion or the deletion. So we say we found, or the data, we observed 3 indels in the sequence we compared. So that's the sentence some people will use. We directly use indels to indicate this stretch of sequence. It's either an insertion or a deletion. And we already talked about frame shift. Then this is just to tell you the same slide as the previous one. Okay, so I'll quickly go through the chapter 1 about all the types of mutations. And I think you have to recognize those terminologies, and we'll use, for example, we use synonymous and non-synonymous almost in every chapters in this class.